Sounds perfect. Hello and welcome to another Agri podcast. For this episode, we'll talk about and talk to Dr. Christopher Grainger. Christopher Grainger is a specialist of genetic markers within the University of Guelph and worked extensively with researchers such as Dr. Chan and others. Is there any greater introduction that you need? Because not many people would know about you.
Yeah, thanks, Brandon. And I know we're recording this. Just one clarification up front. I'm not a doctor. You can call me Mister.
Oh, my apologies. I thought you were a doctor since you wrote down those scientific papers I referenced a couple times.
That's perfectly fine. I get that said a lot, so I'll take it as a nice compliment. But just to be specific about it. Yeah, and we can edit all this right in the final version, right?
Yeah, and plus I don't really edit. I like to keep some of the awkwardness in.
Oh, okay. That's fair. That's a podcast, absolutely. Yeah, so I've been at the University of Guelph for about 20 years now, and I've had a few different roles within the university, but specifically within the plant ag department. I guess you could call me sort of the DNA guy that provides a lot of resources and techniques and genetic, genomic, molecular tools to different areas of agriculture, and certainly plant breeding being one of the main research programs where I provide support and also do research in.
Excellent. So in your lab, what type of genetic markers do you have on your menu, so to speak?
Sure, yeah. So I guess I'll start with maybe for the audience, well, what is a genetic marker, to begin with? And really what we use genetic markers, or what genetic markers are, is essentially a piece of DNA or a DNA sequence that has a known location within a genome or on a chromosome. So that's really important as we use these tools to help with a variety of different applications. So let's say within the breeding side of things, we might use markers for things like DNA fingerprinting, and that could be used for seed purity purposes. Other useful utilities of markers would be when we want to do more basic discovery work. So let's say we want to investigate how much genetic control there is over a specific trait within a crop. And, not only what genomic regions might be impacting those traits, but then also how much genetic, we would say genetic variance or genetic contribution that goes into the control over a specific, trait that we're trying to enhance or breed for.
Can these also be used to determine how much genetic diversity is in a particular population of breeding lines?
Certainly, yeah. So we use markers all the time to understand relationships amongst plant varieties or really any species. And a great analogy to how we understand genetic diversity would actually be in humans. So if I was to take a sidebar a little bit and talk about how we use DNA technologies, let's say in forensic identification or that sort of application. We can use a very limited number of markers actually that provide a tremendous amount of information in terms of relatedness between, let's say, humans and people. And we can use that to develop very specific fingerprints at the individual or person level. And that's why they're used, standard now in, let's say, criminal investigation or paternity testing and these kind of things, because they are so specific down to the individual level. And so if we use that sort of analogy in plant breeding, certainly we can understand to a greater amount the specific genetic diversity that exists in, crop species. And typically within, the breeding hierarchy, you would have what we would call land races, which are essentially the wild version of different crop species. And then as we've done selective breeding over thousands of years, we've created very unique, what we would call germplasm pools, or bred for specific combinations of genes that are favorable for a given growing environment. So the genetics of crops that are bred specifically for Ontario would look very different than genetics of plant varieties developed for other locations, like let's say Brazil or those types of areas. So what we can really do is understand the specific genetics or genetic diversity at a local or regional level, and then compare that to other sources of genetics really worldwide. Because the nice thing about molecular markers and DNA technologies is that it does create a somewhat universal language for understanding genetic variation, in that the genetic code ATGC is ATGC, and it can really lend itself well to handling large amounts of data to compare almost the complete genetic code of different individual plants, populations of plants, and also other sources of genetic material.
Excellent. So you could do this. You can do this to compare how the gene pool genetic variation could change throughout time, like comparing the genetics of, say, 1950 with 1990 or 2012, right?
Certainly, yeah. And so we can really look at how breeders have developed and bred for very specific high performing varieties within a certain geographical location. And what we've typically seen is it really is a filtering process, if we want to think about it that way. So we have all this wild genetics, if you will, if you're starting with a land race, and you wanted to then breed that for commercial application, and you would then be selecting over various cycles, and you would be eliminating most of the deleterious genes. And this might be a good point to make a subtle distinction or definition. When in, let's say, plant breeding, we'll talk about the genes that affect or impact certain traits, but it's also, we have genes, but then we also have alleles of those genes. And so what an allele is essentially a variant. And certainly the last couple of years worldwide with the pandemic, I think most of the general population, has that term variant now well understood within the vocabulary. So we really have been dealing with the concept and the idea of genetic variance for thousands of years in the context of plant breeding. So what we're really trying to do as breeders and using these genetic tools is really help identify the most beneficial alleles of genes, and then combine those favorable alleles through selection, whether that's phenotypic selection or traditional plant breeding, or whether it's through marker assisted selection, where in that case we'll actually have a marker that's either very, very close to a genetic region that we know controls the trait, or nowadays, thanks to tremendous advances in genomic technology, like let's say next generation sequencing, we now have a lot of markers that are actually in the genes that we're interested in. And so then that really makes it quite simple to then use those technologies to help us first identify the favorable alleles, but then also bring those favorable alleles together and then evaluate the effect of those favorable alleles in populations when we're trying to increase whatever trait of interest.
Now, there's many different types of genetic markers,
Certainly.
and such as SNPs and SRRs. Do you use all of those type of markers in your lab, or just a few?
Really, a marker is a marker is a marker. So it just depends on what you're interested in doing and the utility of those given markers. So yeah, throughout, I guess, the last 50 years or so, we've definitely seen tremendous changes in the way that we produce genetic markers, and then also the type of genetic markers that we use. So the very first type of kind of genetic marker essentially was cutting the DNA at specific locations with what are called restriction enzymes. And so restriction enzymes, how they work is they recognize a very specific sequence in the genome, and they'll cut at very specific locations. And so in the very early days of molecular tools, these were one of the first. And so what they found was that different individuals would have different areas of their genomes where these restriction enzymes would cut. And these were, first the most simplest way to have a marker system. Now, as technology has changed so rapidly within the field of genomics, how we generate genetic markers has also changed quite significantly. So I guess over time, we would have, let's say, restriction enzymes as a first type of marker. Then we move to other types of marker systems that would be defined basically on their technical definition. And so we would have things like AFLPs, which would stand for amplified fragment length polymorphism, or RAPIDs, which stood for random amplified polymorphic DNA. As we got more sophisticated with the markers, we eventually found SSRs, as you said, or simple sequence repeat markers. They really became the dominant marker about 20 years ago because they were very easy to use. And most importantly, they were highly what we call polymorphic. And this is really important within any sort of DNA marker system that you want to use. You want the markers to be informative. And what I mean by informative is you want those markers to be able to discriminate the different genetics between different individuals, say individual plants or germplasm sources. Because a marker will either be what we call monomorphic, which is it'll produce the exact same data across all individuals, which really isn't that informative, for plant breeding purposes, because we want to know the genetic differences, or again, the alleles of different markers and genes. And so we really want a marker system that contains good polymorphic information. And what I mean by that is it's able to tell the allelic difference between individuals at that marker. So I'll maybe use disease resistance as a great example of a trait like this. So let's say we want to introduce a new source of disease resistance from some line. So the different varieties or the different individuals, they'll have the gene for disease, resistance, but it's really the alleles where you might have one that's carrying the resistant allele and another one that's carrying the susceptible allele. So in that case, what we want is a marker that's going to be able to distinguish between the resistant source and the susceptible source. And so SSR started out as really the first sort of high throughput main marker system that really was used in wide application in many, many different fields. DNA forensics, as I was talking about earlier, that's the marker system that we use to identify every human on the planet. Likewise, in plant breeding, they were used very early on because the nature of an SSR marker is that it's a small little repeat sequence. So let's say I have a sequence of AAT. And how that works is that AAT sequence is repeated as that unit many times. And how those markers work is that it's the difference in the number of that repeat between individuals. So for example, I'll have a marker that has AAT, and that might be repeated 20 times in an individual that is carrying a gene for, let's say, yield. Whereas, let's say high yield, whereas another individual might be carrying that same repeat, but it's only 10 repeats. and they might be carrying a negative allele that affects yield. And so really what we're trying to do with the SSRs is use those to help select for the proper allelic variants for different traits. Now, those were sort of a first generation marker because those types of repeat sequences are found everywhere throughout the genome. So that's why they were very attractive, because these sequences are found at high density across all the different chromosomes of a different crop, or any species for that matter. Really, when we shift now to SNP markers, which are single nucleotide polymorphic markers, that's the official definition of them. Really, what we're talking about is now we've shifted everything to the actual differences in the DNA sequence code. And a SNP would essentially be the difference at a specific, what we would call DNA base or nucleotide. So the genetic code is the genetic code, ATGC. And so a SNP marker is really just the difference between an individual carrying the A nucleotide versus a T nucleotide, let's say. And the main reason why those have now become the dominant marker system is through the technological advances such as next generation sequencing, where now you can sequence almost an entire genome, and the amount of information you get from that is tremendous. And you're just able now to use this data that's produced to then cross-reference it to what we call reference genomes, which have now been produced for most of the economically important food species. And now it becomes a very, quite nowadays, simple exercise to understand genetic variation or DNA sequence variation at a very large scale at the genome side, at the genome level, between different, breeding lines, populations, and then also how we use this type of transformational data to help improve breeding strategies or develop new breeding strategies.
So just to make sure I'm understanding this correctly, the SNPs can be used to gather a lot of genetic data to make a sort of like a genome catalog for individual breeding lines showing, or individual lineages of plants, to showcase what traits are in what lineage and how they're inherited.
Certainly, yeah. I would say I can give a couple of examples of very applied, aspects of markers. So yes, that first one about understanding, say, genetic diversity at a much greater level. I'll use another human analogy. I think a lot of people have heard of companies like 23andMe, where people will send their spit sample away to a company that will sequence, and they'll tell you a lot about your specific heritage, or how much European ancestry or whatever ancestry you have within say your own lineage. And that's informative to understand, different, say, genetic diversity aspects. I would say, if we're swinging back to plant breeding, there's much more application for developing new varieties or new products if you're a seed company with the technology. So let's say 10 years ago, we might use markers for what we would call classical marker assisted selection. So that's where we would have a breeding population, and we would then genotype that population at a few markers to understand, what alleles we want to maintain for the next cycle as we select, and as we select on those populations, and we start to breed for more elite, material. One thing that's really come on the scene the last couple, maybe about 8-9 years now, is the concept of genomic selection, which is different than marker-assisted selection. So within genomic selection, what we're actually trying to do is leverage all the information that we have now. Instead of looking at things from 5 or 6 genetic markers, we might use the combined information of 50,000 genetic markers scattered across all the different chromosomes. And what we can do with that is actually do predictive selection based on the genotypic makeup or the genetic code of different lines, and really expedite favorable genome-wide combinations of alleles and markers to help breed, enhance breeding, and also reduce the time it takes to get new varieties to market. The other thing I'll say about markers is that they sort of fall into two general categories for application. One is just routine applications. So let's say I'm working in a seed company and I just want to maintain seed purity of a line, or I want to use them to do fingerprinting, genetic diversity, those kind of analyses. The other way that we use them is more on the research and development or discovery side. And in that case, what we're doing is we're creating populations of breeding material, and then we're using these technologies to help actually do the discovery side and identify or discover the genetic regions or the genes or the combinations of genes that actually have a impact or govern a certain phenotype or a certain trait. So there really is sort of two sides to genetic markers. Some are just routine commercial application, while other ways we're actually using them to discover, or make new discoveries within plant breeding and genetics.
Thank you. We covered a lot. So just to clarify, a whole genome can be utilized with all the markers to create a good analysis of what type of traits will be passed on from one parent to another. And you can use genetic markers either to determine the purity or discover new traits.
Yeah, certainly. And those are some primary usage of genomic technologies within plant breeding. It really is always the combination of two different things. We've got the genotype or the genetics that impact a trait, and then the other side is environment. And those two things are always tied together. And if you want to make progress in breeding, it's really the combination of both that is critical to advancing, any new, variety development or crop development strategy. And so a lot of times what we can do is we can't really control the environment, but we can control the genetics in how we breed. So it's that combination of understanding the right genetics within a certain growing region or environment that becomes critical. So that's really where we see the most impact, is when we're utilizing tools in that way that they're combined with everything else we're doing in a traditional breeding sense.
Thank you. So we can use this to determine, does the genetic markers also include whether or not each trait is dominant or recessive as well?
There are marker systems where that absolutely does work, and that's part of what we call the genetic architecture of any trait or breeding objective. And that gets into classical genetics, where certain traits are controlled, or the underlying genetics of the trait are controlled by classic Mendelian laws, where we've got dominant, recessive. We also have other genetic terms that we use in plant breeding, such as epistasis. And the definition of that is where we have multiple genes, and some genes influence the regulation or the expression of other genes, then control the expression of the trait. So that can be a major source of research, is understanding, the genetic architecture of different traits. So I'll give an example. So something like yield within any crop species, it's not exactly like there's 2 or 3 yield genes. Yield is a byproduct of, hundreds, if not thousands of genes, working together. And then also those genes being expressed in different environments. So really, when we talk about how we use or understand genetic markers within different traits, it does have to do with the complexity of the trait that you're wanting to use the markers in. And so that's where something, certain traits are controlled by a few genes. We call those, major gene traits, and that you could use a few markers because the major genes have a big effect on the final, phenotype of the species. Whereas other times with more complex traits, you'll need not only to understand the influence of many different genes, but also how those genes interact with the environment that you're trying to breed for.
Thank you. So to broaden that into other topics, QTLs are, from what I remember, a genetic marker that could be used to determine where a certain level of genes that code for certain traits dwell. Is
Um,
that correct?
So that would be certainly on the discovery side of things, and when scientists and breeders and researchers are trying to discover, what are the genes. So let's say you're working on a trait that really hasn't been investigated too much, and what your objective is to try to understand the genetic component of that particular trait. And certainly the idea or the concept of QTLs, which the definition of a QTL is called a quantitative trait loci or locus. What a QTL is really a statistical association between a area or a genomic region on a specific chromosome and how much of the overall phenotypic variation is due to the genotypic region and not due to other sources or other parameters. So we would break that down and say, as an example, the total, let's say, oil concentration, if we were talking about, say, an oilseed crop such as soybean, and we had we wanted to increase the overall oil concentration. And so we would say how much of the oil concentration or trait is controlled by genetic factors versus environmental factors, or the combination or the interaction of both of those factors. And so that's where the greater percentage of genetic control over a specific trait, the more we can utilize that in a marker system to help breed for new advances in traits.
And it can be also used for disease resistance and yield and other stuff like that, correct?
Really, it's unlimited. The markers are there to be used as, I would say, definitely genomic or molecular tools to help understand the genetic components of any trait development.
Excellent. So bringing it back to exploratory versus maintenance.
Mm-hmm.
What type of what type of markers will be used for each one?
Really, the marker systems, I would say, as a clarification, would be those are the technologies. And so using those technologies would be very similar to any other technologies that are used in agriculture. So, if you're going to use drones to take images, or you're going to use, other types of analysis like soil health measurements, or, it's really an artifact or it's a part of the technology, which are marker systems. So typically the marker systems would be used for genotyping purposes. And so then the tools are the tools. It's really what application you're wanting to use within those tools that would be the decision making step in what kind of marker system you might want to use. I will say, because of massive technological changes on the DNA sequencing side of things within the field of genomics, it's very quickly shifting from, let's say, genotyping with older technologies such as SSR markers, to now the dominant form of genotyping is simply by sequencing a genome within that crop species. And there's a reason, there's two aspects of that. One is the cost to produce this kind of data has fallen dramatically because of the technology. And then the other side of it is we just have better computational technology and abilities to handle these kind of, or this amount of data, and use it more routinely within, say, breeding programs or whatever.
And I think that's everything. Thank you.
Okay, that was great. Thank you for having me on.
It's been a pleasure.